again and back into CPC. Thom is a professor in the Department of Anthropology. He's a faculty fellow at the Institute for the Policy Research, and he's director of the Laboratory for Human Biology Research and director of Cells to Society, all at Northwestern University. His work is truly biosocial, and he focuses on the relationships between society, biology, and health with a life course perspective. A lot of his work deals with issues around stress and immune function. And one of the things that Thom is very well known for and a leader in the field of is the development and use of minimally invasive methods within population research. And it sounds like that's what we're going to be hearing about today as well. He uses these methods in ongoing research in Bolivia, in Ecuador, with the Cebu project in the Philippines, as well as applying these questions to health disparities within US-based research. His work has been funded by the National Science Foundation, National Institutes of Health, Werner Gren Foundation, published in all of the leading social science and medical journals. And he was recently elected member of the National Academy of Sciences and is a fellow of the American Association for the Advancement of Science. So it's a real pleasure to have Thom here today. And so let me turn it over to him for the talk and just give everyone a reminder. You're more than welcome to put questions in the chat or to participate by turning on your camera and turning on your mic at the end of the talk for questions as well. So thank you, and looking forward to hearing your talk.
Thanks, Amanda. Thanks for the kind introduction. It's great to see you all, albeit virtually. I was just saying that I'm really disappointed I couldn't make this visit in person, but I'm committed to coming back. So you'll see me all again. You'll see me again before too long, I'm sure. CPC and UNC are very special places for me. Most of you know, or at least people of my cohort and know that I was a trainee at CPC in 1999 to 2000, and that experience has had a huge impact on my career. And it's in many ways the gift that keeps on giving, because as you'll see in my talk, I have some new collaborations with people at CPC that I didn't work with during my postdoc, but now I do. So it's a pleasure to be here with you all. This is a big picture talk. I'm going to start by sharing my screen here. And it's a great opportunity to share with you my current thoughts on where I think we have been with respect to this integration of biological and social science approaches and methods, and where we can go, next steps. And this is an exciting place to be, and CPC and UNC are at the leading edge, particularly in this training space. So I'm excited to share these ideas with you and get your input and collectively think about where we might want to go and what are the most productive future directions to have the biggest impact on population health. So it's a big picture talk, but you'll see I'm going to get into the weeds on a couple of things every now and then. So please, add questions to the chat. I probably won't get to them in real time, but obviously at the end, and there should be plenty of time for conversation. All right. Let's see here. This is not advancing. Try again. Oh, there we go. Are you all seeing that? Did I lose you?
Yeah, we lost the slides.
Lost the slides. Okay, I'm going to try to share again. Okay, are we back? Slideshow? We're good? Okay. All right. So let's start with the Human Genome Project. In 2003, the human genome was sequenced, cost $3 billion, and there's no question that this was a tremendous technical achievement. But there was no shortage of hyperbole following the announcement that we now had a complete map of the human genome. And so, for example, Francis Collins here on the left, now director of NIH, writes that, or spoke publicly that it's humbling that we now have our own instruction book, previously known only to God. A counterpart in the UK wrote that we now have the set of instructions to make a human being. And then another science minister in the UK also said that we now have the possibility of achieving all we ever hoped for from medicine. Now, fast forward 5 years, and it started to become apparent that things were a bit more complicated. There was a lot of head scratching in the scientific community around what was called, or still called, the case of the missing heritability. Now, prior to sequencing of the human genome, twin studies, other family-based research designs suggested that many complex traits and diseases were highly heritable. But when the human genome was finally mapped and the actual genes were nowhere to be found. Fast forward 10 years, a decade after the sequencing of the genome, and things are just getting worse. And people are now starting to ask publicly whether the whole project was worth it, whether it was a failure. So Nicholas Wade, now discredited, largely discredited science report from New York Times, I'll get back to him in a minute. He wrote that 10 years later, the genetic map yields few clues. A few new cures. In The Guardian, we have the headline, "The Failure of the Genome." And then my favorite one, Slate, down here below, blogging the human genome was the project of failure. According to his published DNA sequence, James Watson should be blind, deaf, and have a tiny head. Why doesn't he? Well, it still gets worse in the sense that adding insult to injury, the Human Genome Project led to a dramatic downsizing of the human genome. So when the project started, and before, when I was in college and graduate school taking biology courses, I was taught that there were 120,000 genes in the human genome. But as the sequencing started to come online, that number came down precipitously. And now we have a pretty precise count of the number of protein coding genes in the human genome, and it's around 21,000. Way down from a figure of more than 100,000. And as a point of reference, we have 21,000 genes in our cells. That's about 10,000 fewer genes than the tomato has. So a tomato genome has 31,000 genes. We have 21,000 genes. Now, in response to this finding, Nicholas Wade writes in the New York Times that the tomato and the potato contain so many genes does not mean that they are more sophisticated than people. And he writes this with no sense of irony or sarcasm, at least than I can tell. And when I read this, I wondered, did he really feel the need to affirm that humans are more sophisticated than tomatoes? In one sense, there's an existential crisis here, which I think is probably not surprising if you think that the origins of what it means to be human trace exclusively to our DNA. So what can we learn from a downsized human genome? In my opinion, this discovery may be the most important scientific contribution of the project to date, because it forces us to confront the fact that the secrets to human nature, the cures to complex diseases, they're not locked in the genome. The case of the missing heritability will not be solved with approaches that privilege simplistic models of genetic causation. And this is where we come in. As developmentalists, social behavioral scientists, population scholars, we've long emphasized the complex interplay between genes and society across the life course. The failure, quote unquote, of the Human Genome Project actually presents an opportunity. And I think we've arrived at a unique intellectual moment when the population sciences can make important contributions to our understandings of human biology and its complex multi-level determinants. And so here are a handful of models. I'm sure these are familiar to many of you coming from different disciplinary traditions. Subdisciplinary perspectives and traditions, but they all emphasize the nested and contextualized nature of human biology and health. Now, importantly, these kinds of approaches have been around, a biosocial approach have been around for probably more than 100 years. So it's worth noting that one of the first examples, at least that I'm aware of, was by W.E.B. Du Bois, who used morbidity and mortality data across cities in the regions in the US to underscore the fact that social and economic conditions, not inherent biological difference, explained higher rates of disease among African Americans, particularly in the US South. And then working at the same time and occasionally in conversation and collaboration with Du Bois was Franz Boas, who's recognized as a founding figure of American anthropology. And he used anthropometric measures of skull size and shape to show that children born in the US, regardless of their ancestry, had skulls that look more similar to each other than to their immigrant parents. So this work challenged prevailing typological assumptions that cranial form was a fixed and inherited marker of social identity. Now, fast forward more than 100 years, and more recently, in the 21st century, we have volumes and meetings that have explicitly promoted the integration of biological measures into population-based research and biosocial research more generally. In 2003, National Academies produced this volume, Cells and Surveys. And interestingly, the subtitle here is a question: should biological measures be included in social science research? And there was a lot of concern among social scientists, particularly those with long-term cohort studies, that the integration of these biological concepts and methods and measures might turn participants off, lead to high rates of attrition and bias sampling, and so on and so on. 5 years later, the follow-up volume, Biosocial Surveys, no longer poses this question. The horse was out of the barn at this point in time. In part through a lot of encouragement from NIA, other funding sources, there was widespread application of biological measurements in the context of social science and population-based surveys. So partly in response to this, and with an opportunity from the Russell Sage Foundation, Kathy Harris and I collaborated on editing a volume on biosocial perspectives, put together a nice set of papers, and we wrote the introduction for that volume. And what we realized early on is that people widely use the term biosocial, myself included, but we don't often define it. So we strove in this introduction to provide an overview, a bit of a history of the field, where it's at, and provide a definition, and then also suggest productive steps moving forward. And so a lot of the talk at this point forward is going to draw on this chapter, and I'm indebted to Kathy for helping me think through these ideas. So we define in this introduction the biosocial as the dynamic and bidirectional interaction between biological phenomena and social relationships in contexts which constitute processes of human development over the life course. And so we have a model that begins here with the social world. So we have sets of nested and mutually reinforcing contexts and experiences at multiple levels, including interpersonal relationships, neighborhoods and schools, and broader social and political economic structures and forces. And in short, these can be understood as processes that exist outside of the body. Now, similarly, the biological can be understood as nested and integrated processes and structures inside the body that guide the growth, reproduction, and maintenance of the soma from conception to death. Now, we can productively study the human experience within each of these levels, and a lot of our academic disciplinary structures are built around understanding these contexts and processes within each of these levels. But what we're trying to promote here is a biosocial approach that recognizes that the biological and social are in fact mutually constituting, particularly within a life course perspective. And we're trying to deliberately blur the boundaries between phenomena inside the body and outside the body. A biosocial approach implies that attempts to understand one without the other are incomplete. So where are we now? I think we've arrived at an important juncture with unprecedented opportunities for innovative, high-impact, population-based biosocial research. The acronyms here on the left are familiar to many, if not most of you, I'm sure. They stand for large population-based surveys designed primarily by social scientists, and each study has a diverse and representative sample with thousands of participants. Each includes rich measures of context, experience, and behavior. Most contain multiple measures across the life course. Some focus on aging, others on earlier stages of the life course. Some are now intergenerational in their emphasis. And they all have important human capital outcomes across multiple dimensions. Now, what sets them apart from standard social science surveys is that they all include direct biological measures, including measures of anthropometric status, measures of growth, development, nutritional status, and history, physiological measures that tap into various metabolic, immune, cardiovascular systems, as well as genetic measures, including measures of gene sequence, as well as gene function, like methylation and gene expression. So it's a really unique opportunity. Now, what sets them apart from most clinical research is that these are large, representative, and diverse samples with rich measures of context. So it really is, I think, a unique opportunity for us to think about how we can bring the bio into population-based research. So we should ask ourselves, what can this do for us? How can this advance our broader projects? And I think one key point is that biological measures allow us to document the health consequences of contextual factors we care about, like socioeconomic stratification, structural racism, social relationships, neighborhood quality. Biomarkers give us tools to better understand how environmental factors get under the skin, quote unquote, to use an often used and perhaps overused phrase, how these contexts become embodied and shape our health, and through which specific physiological and biobehavioral pathways they connect experience to health outcome. And for better or for worse, objective biological data related to clinical outcomes in particular may be more effective than subjective health measures in mobilizing the attention of policymakers around important social issues that we care about. Biological measures also provide insight into pre-disease pathways that allow us to identify individuals at highest risk for the future development of disease. So with this information, we can evaluate the potential health impact of social context earlier in the life course, rather than having to wait for years or even decades to see who is in fact at greatest risk for morbidity and mortality. Now, much of the work integrating biological measures in population-based research draws on epidemiological frameworks, and I think that's good, but we also have to be wary about just doing bad epidemiology. As social scientists, I think we have an opportunity to use these measures a little bit differently and to think about how biological measures provide insight into the quality of the environments that people inhabit. And it allows us to identify which exposures must be most toxic and for whom. Also allows us to identify resiliency factors in the face of adversity. And it also allows us to evaluate how biological processes might actually inform selection into different types of experiences and contexts, particularly within a life course framework. So where are we now? Overall, I think we can reasonably summarize the current MO of population-based biosocial research as borrowing clinical biomarkers and applying them in population-based settings using prevailing conceptual and analytic frameworks. And we've got a lot of purchase so far. I think we've done a lot of great work in this space. I would like us to move forward in what I'm calling Biosocial 2.0 and encourage us to pursue a more ambitious goal. And that is to challenge prevailing biomedical frameworks by broadening scientific and public understandings of human biology, development, and health. Now, here's why I think this is important. Biomedicine, from which we tend to borrow not only methods, but also broader conceptualizations of human biology, is defined by explanatory models that locate causation inside the body. So here are the quotes that I shared earlier following the initial sequencing of the human genome in 2003 that underscore the primacy of DNA as a causal force, right? But it's not limited to DNA. We also have sentiments like this: the natural inclination of medicine is to isolate a single factor that is most responsible for the observed behavior. For infection, the target is the pathogen. For cancer, it's the tumor. For gastrointestinal bleeding, it's the bleeding vessel or ulcer. It's a very reduced and isolated approach to understanding biology and health. Now, you might think that epidemiology might get outside of this trap and get outside the body a bit, but many, if not most, epidemiologic studies focus on individuals and individual risk factors. And I like how Len Syme sums us up here. He says, this research might more properly be seen as clinical research in large groups of people. And this privileging of explanations inside the body is not limited to practitioners of biomedicine and biology. It reflects and reinforces broader cultural models regarding the primacy of genes and other individual level factors as the most important determinants of health. So for the past several years, I've been teaching an undergraduate 200 level course in basically the social determinants of health. And on the first day of class, I have the students fill out a survey. And I ask them a question. I ask them, what do you think is the most important determinant of one's health? Some people are healthier than other people. What do you think are the most important factors that determine a person's health? And they generate a list. And this is a simple word word cloud from the pre-class survey. And as you can see, diet, genetics, exercise, these things pop out. And these are all individual little attributes. So the genes are inside the body, but diet and exercise, these are like personal responsibility, healthy lifestyle choices, quote unquote, quote. These are the most important determinants of one's health. And then my own university, Northwestern, likes to emphasize how it is an interdisciplinary institution that blurs boundaries between departments and schools. And so here's a campaign ad that talks about, it's hard to see at the bottom, but Northwestern is micro and macro, tradition and innovation, local and global. And then the catchphrase is, and is in our DNA. And they use DNA because they want to emphasize the point that it's so important and essential, it's embedded, and it's immutable. Right? And it is a causal force that makes us this unique institution. However, when explanatory models privilege forces inside the body, it's more difficult for us to conceptualize human biology as a social and contextualized biology. But humans are long-lived social creatures. And when you get right down to it, I would argue that the two most important determinants of human physiological function and health, the ones with the most explanatory power, are actually the quality of an individual's social relationships and an individual's rank in society. Now, this is not news to most of you, I'm sure, and most of you are familiar with foundational work by Jim House and others. There's just one example showing that as people become more socially integrated and connected, their risk of mortality goes down. And so now we have more contemporary versions of this story by Yang et al. With Kathy Harris as a senior author on this paper, showing that among young adults in AdHealth and older adults in HRS, individuals who are more socially connected and integrated have lower risk of cardiometabolic disease, as indicated by various clinical indicators. And then now we're also starting to get inside the genome. This is an important early paper from Steve Cole and John Cacioppo showing that individuals who are socially isolated have really different profiles of gene expression than individuals who are more socially integrated. So the fact that our bodies have evolved down to the level of gene function to be sensitive to social inputs further underscores the social nature of human biology. And of course, it's now widely known that social rank is a fundamental determinant of human health. Early work by Nancy Adler and colleagues have helped put this problem on the map, the problem of the gradients, higher levels of socioeconomic status associated with lower levels of mortality across almost all causes and all places. And of course, there are now thousands of studies on various physiological indicators of disease risk like CRP, blood pressure, lipids, et cetera, showing education, wealth, and income gradients in disease risk as indicated by these physiological indicators. And now we're also starting to get inside the cell and look at patterns of gene expression and methylation in relation to SES. And this is an example I'm going to get to in a few minutes. So moving forward with Biosocial 2.0, I suggest we continue to attend to developments in the clinical sciences and borrow as needed. But I would like us to challenge, I'd like to challenge us to do more, to broaden scientific and public understandings of human biology. In order to do this, I think there are three important areas that we can make important contributions. There are probably more, but these are the three that I'm going to cover today. So the first is to innovate methods that allow us to study biology and health in quote unquote free-ranging humans, move away from a exclusive clinical emphasis on the study of human biology and health to get out there in the real world where we can foreground the experiences and the context that we know matters to shaping human physiology and health. We can use biology to document the impact of social context and experience. I'm going to provide an example of that. And then I think we can do good work, important work in challenging simplistic understandings of biological causation. And I'll end with an example of that as well. All right, what am I doing on time? Okay. Measuring the body in free-ranging humans. So UNC has really been an innovator in this space, and I've been privileged to work with people in AdHealth and other studies in developing some new ways to think about measuring human biology and health in non-clinical settings. So now the range of wearables, smartphone devices is increasing rapidly, allows us to get measures of physical activity, sleep, cardiovascular function. We have a wide range of anthropometric tools to measure nutritional histories and status, cardiovascular disease risk. We can look at multiple measures of reproductive function and stress and infectious disease and saliva and urine. And of course, blood, which tends to be the clinical standard for measuring many physiological processes and health outcomes. We can do a lot with a single drop of blood, particularly with the technological development of more sensitive assays using smaller volumes of blood. So we can use point of care instruments where we provide a measurement at the point of collection in someone's home or community-based settings. We can put blood on slides, we can put it in capillary tubes, and then most of my work is focused on the use of dry blood spots, which I'll talk a little bit more as a way to get blood in the field and bring it into the lab. I don't have time to talk about this, but I'm very excited about some work I did right before the pandemic in collaboration with Greg Miller, who's a psychologist who does a lot of work on stress and inflammation. He runs a lab where they routinely bring people in and they'll draw venous blood, and they'll take that blood and they'll pulse it with an antigen, a stimulant that activates inflammation, and then they'll add glucocorticoids and other things that turn it down. So it becomes a nice experimental system for activating inflammation and revealing the processes that regulate it. It generates a lot more insight into inflammation and disease risk than just a single measure of CRP, for example. Well, you can really only do that in the clinic or the lab. Well, we miniaturize that whole process where we can, in a couple of drops of blood and with a portable incubator and putting the blood on blood spots, we can do this in the field now. And that all got shut down by the pandemic, but we're starting to think about rolling that out in the near future. So if that's something that anyone is interested in, we have a paper validating this. I'd be happy to talk about it more as part of this talk or offline. All right. So field-friendly methods for studying biology in free-ranging humans allow us to get out of the clinic where we can foreground the contextual factors that we think are determinants of human biology and health. This also allows us to recruit more diverse and representative samples, engage communities in formulating the questions and collecting the data and interpreting the results. It also asks us to ask novel research questions that are facilitated by methodological innovation. All right, second point. Using biology to document the impact of social context and experience over the life course. And I'm going to provide an example on some research we've been doing looking at the origins of social inequalities in health over the life course. All right, so here's the Adler figure. We know that in adulthood, socioeconomic status is associated with morbidity and mortality. And most of this work has focused on context, social relationships, behaviors in adulthood, how they may shape the function and the regulation, the function of various tissues and the regulation of biological systems to put some people at higher or lower risk for various diseases. But we also know, because of large body of research in the developmental origins of health and disease, demographic research on the long arm of childhood, that context, social relationships, behaviors earlier in the life course have an impact on these organ and tissue systems and help establish regulatory set points for biological systems. And in fact, there's good reason to think that these contexts, these environmental exposures may be more important earlier in life because these systems are coming online and there are sensitive periods of development during which environmental inputs may have lasting and disproportionate impact on physiological and function later in life. So what I'm going to talk about right now is a couple of studies we've done on C-reactive protein, key biomarker of inflammation, well-established risk factor for predicting all-cause mortality, as well as cardiovascular disease mortality in particular. So it's now used clinically, epidemiologically. Higher levels of chronic inflammation indicate higher risk independent of other risk factors like blood pressure and lipids. And in a recent paper, pretty much hot off the presses with Kathy and Jess Meyer, trainee that you all know and is in the audience today, former graduate student here at Northwestern. And then Stephanie Koning is a postdoc working with me here who is now on an F32 fellowship. Together, we did an analysis of body mass and chronic inflammation with AdHealth data. And what we show is that this is nothing new, but there are large education gradients in chronic inflammation. What is new with the AdHealth data is that the overall level of chronic inflammation is quite high, and that we're seeing high levels and large gradients in younger adulthood, whereas most of the research on C-reactive protein chronic inflammation and health outcomes is on older adults. And we also find that there are very big differences in BMI in the cohort, and that these differences in chronic inflammation are explained in large part by the high levels of obesity. Now, previously, in 2014, I did an analysis with AdHealth data, wave 4 data, where we looked at birth weight and breastfeeding duration as predictors of CRP in wave 4 of AdHealth. And this was motivated by the developmental origins of disease idea, which at the time had been doing a lot of work on cardiovascular disease outcomes like blood pressure and lipids, much less on inflammation. So we were able to show that individuals who were born with a higher birth weight had lower levels of CRP, lower chronic inflammation in young adulthood. And then interestingly, and this was among the first to show this, individuals who are breastfed for longer periods of time also had lower levels of chronic inflammation in young adulthood. And these findings have since been replicated in other cohorts and other contexts. We also did some sibling difference models. So at least in my mind, these are important and likely causal effects of early environments in infancy on the regulation of inflammation later in life. And in another new paper, this one is just with me and Stephanie, we're trying to put these things together. So we know that in adulthood, socioeconomic status is related to chronic inflammation, and part of that association is due to body mass. And we know that lower birth weight and breastfeeding duration are associated with BMI as well as chronic inflammation. But we don't know the extent to which this adult gradients, these adult inequities in chronic inflammation, might actually trace back to differential levels of birth weight and breastfeeding in infancy, which is also socially patterned in the United States. So individuals with lower wealth, lower levels of education are more likely to give birth to lower birth weight babies, as you know, and rates of breastfeeding are also lower among lower educated and lower income individuals. And this is due largely in part to the fact that we do not do a very good job in the United States of supporting families in this process of generating a family. And breastfeeding in particular is a hard thing to do. And in many ways, it's a privilege to be able to do it, and particularly to be able to do it for long periods of time. So it's socially patterned in the United States, unfortunately. So within a structural equation modeling framework, we tried to put these pieces together to ask the question, to what extent are education or SES gradients in adulthood and chronic inflammation, can they be explained by or traced back to differential levels of birth weight and breastfeeding in infancy? And we have various controls that we, of course, apply to our models. We measure CRP in wave 4, blood spots from Ad Health. We have a composite socioeconomic status indicator. It includes education, income, neighborhood poverty, perceived SES. We have BMI, and we just look at, for simplicity, if an individual was born less than 2,500 grams, low birth weight or not, and then we have a threshold of 3 months breastfed or not, and that was based on our prior research. And here's what we find. This is, I'm going to show you a sort of a stepwise pattern of associations. That initially we document a strong SES gradient in chronic inflammation, so no surprise here. Individuals who have higher SES in young adulthood have lower levels of chronic inflammation. And then if we put in our control variables and also parental education, we attenuate that gradient a bit. We explain a little bit of it, but there's still a strong gradient there. And then when we put in low birth weight and breastfeeding duration, the gradient virtually disappears. We can explain 83% of the explain away 83% of the association between socioeconomic status and CRP in the Ad Health cohort if we assume that everyone was not born low birth weight and that everyone was breastfed for 3 months or more. So it's sort of a modeling exercise here. So there are obvious limitations in a observational study where we're doing basically a modeling exercise. But to me, this is compelling evidence, or at least promising initial evidence in support of the hypothesis that environments in infancy influence chronic inflammation later in life. And if we can enact policies to support mothers and families to improve birth outcomes and promote breastfeeding, we can have a substantial impact on reducing social inequalities and chronic inflammation and CVD risk in the next generation. Okay, so that's one example of how we can use biology to document the impact of context and experience, and particularly to highlight the importance of the life course, which, as many of you know, is not really emphasized in clinical and many epidemiological approaches. I'm not going to go into detail here, but I think it's also important to say again that biomarkers can be used as ethnographic tools. They give us some insight into the quality of social environments in which people live. And these tools can also be very useful for policy evaluation, in the sense that we can evaluate the impact of some intervention in the near term rather than waiting a long period of time for clinical disease to emerge. And we can also perhaps evaluate unintended consequences of, say, a social policy that might not be intended to have an impact on health, but may in fact have that. If we can detect that, that might be an important part of cost-benefit analyses. All right, I'm going to move on to my last example here, and then my last point here, and then we should have time for a conversation. So the third point that I'd like us to consider, that I think we in particular as social scientists are in the best position to pursue, and that is challenging simplistic understandings of biological causation. Now, for those of us who are interested in understanding how experience becomes biology, there's an emerging opportunity to reframe our understanding of how genes matter to health. Now, most of you, I'm sure, all of you have heard of the term epigenetics. But it refers to a broad class of biochemical processes that lead to stable changes or differences in gene function in the absence of actual changes in the inherited gene sequence. Now, epigenetics has generated a lot of excitement because it presents a plausible biological mechanism through which our bodies remember experiences early in life that then go on to shape human development, biology, and health later in the life course. DNA methylation has been a major focus of human research, and it involves the binding of methyl groups to CpG dinucleotides in gene promoter regions. And the methylation limits access of transcriptional machinery to the promoters and thereby reduces the expression of these genes. Now, that's the canonical model. It's not quite that simple, but this is the model that guides a lot of research, and it's largely true in most contexts, but not all the time. So for the past 5 years, I've been involved in a series of studies with measures of DNA methylation as well as gene expression, including this one, where we asked whether we could detect molecular signatures of socioeconomic adversity in the epigenome. And we did this by taking advantage of DNA that had been previously collected as part of the Cebu Longitudinal Health and Nutrition Survey. And when I first came to CPC, after earning my PhD at Emory University, I worked with Linda Adair, and she introduced me to CLHNS, and I've been working with it ever since. And in the context of this study, or some background on the study, the participants were pregnant women in 1983, 1984, who were recruited into the study when they were pregnant. They gave birth to their babies. Those individuals have been followed up their entire lives, and we have a more recent follow-up in 2014. And we have rich measures of context, experience, caregiving, neighborhood quality, beginning in gestation through infancy and childhood, and then again, of course, in adulthood. For the purposes of the study that I'm going to talk about now, we looked at the SES, the household level SES environments in infancy and childhood as a measure of, as measured by parental education, level of household income, and level of assets. And then we also had comparable measures of the individual as they were an independent adult in adulthood when we measured, when we collected their blood. In 2005, we have these DNA samples, and they were interrogated for genome-wide DNA methylation using the Illumina 450K array. There was now a new iteration of that, but that was what was available at the time. And here's what we found. So this is called a volcano plot. And it has on the y-axis p-values, which makes me profoundly uncomfortable. I don't like really this way of doing this kind of work, but it is the standard. And what we show here is we compare individuals who in childhood were born into low SES environments, and then as adults also were low SES, in comparison with individuals born into higher SES households and then ended up as high SES adults. We have 1,777 CpG sites with increased methylation and 769 CpG sites with reduced methylation that were significantly different across these two groups. And I can explain why we compared the extremes later if there are questions, but this is how we did it initially. So we have more than 1,500 genes that were in some ways differentially methylated in relation to our measure of socioeconomic adversity. And then we can do bioinformatic analyses, gene ontology analyses, which help us identify what are the themes, the common themes in the genes that are represented across as differentially methylated across these two groups. And as it turns out, measures of immune function, measures of skeletal development, and nervous system development all sort of pop up. I don't put a lot in that, and actually that wasn't the most interesting finding to me in the study. The most interesting finding to me was in the context of our understanding of the number of genes in the human genome, that 1,500 genes were differentially methylated. The function of 1,500 genes in the genome was changed by virtue of being exposed to low socioeconomic status, or being exposed to poverty. That's nearly 10% of the genes in the genome were impacted at a molecular level in relation to this measure of socioeconomic poverty, socioeconomic status. That to me was compelling evidence for the social nature of biology, the importance of socioeconomic status leaving an impact on our biology. And so I was enthusiastic when the paper was accepted. I reached out to our press office. I wanted to do a press release, and I thought this is an opportunity to sort of shape the narrative, make the point that that genes are not immutable features of our biology. They're fixed at conception, sure, but really what matters is expression. And our experience in the world shapes the likelihood of expression. So we need to think about the genome a little bit differently, as a more dynamic substrate and not as something that is static and fixed and deterministic. So that was great. And it did get some nice coverage, including in the Chicago Tribune. Poverty plays a role in DNA structure changes. So there were a handful of articles, mostly by good science journalists, who picked up on that theme, and I was very happy with that outcome. But then there was a lot that started to come out in other news sources and in exchanges that I had over email with people that made me realize that there was still there's still a lot of work to be done here. So here's an exchange with someone who wanted to know if it would be correct to conclude that the effect of poverty on genes is harmful. And then I had an exchange, with Christian Cooper, who has written a nice series of articles about poverty and growing up in poverty in the South and what that is like. And he cites epigenetic research to document how poverty is a disease that affects your body in a permanent way. And it has lasting and deterministic effects on your life chances, basically. Now, for those of us like me and you all who understand human biology as contingent and contextualized, research into the epigenetic mechanisms of developmental plasticity seems to provide us with opportunities to challenge the prevailing emphasis in society on simplistic genetic determinism. But we can't necessarily control how our findings are interpreted. And it seems, at least to me, that we have to be careful about replacing one form of reductionism and determinism with another. And this is also a critique that I and others have made around the developmental origins of health and disease hypothesis that really focuses on low birth weight leading to higher risk of disease later in life. And that becomes a new causal and deterministic force. If you were born low birth weight, you're done for. Well, that's not true. It's not that simple. But these are the ways these kinds of findings get consumed. So I do believe that it's important for us to do the hard work of challenging simplistic understandings of biological causation, but there's still a lot more work to be done here. And I think this is something that, I personally am going to need some help thinking about how to do better. But to end on a more optimistic note, as I said earlier, the students in my health inequities class at the beginning of the course, the beginning of the quarter, privileged genetics and other forces inside the body and the brain as the most important determinants of health. But at the exit survey at the end of the class, it's clear that their minds are open to being changed, and their responses at the end of the quarter make me much more optimistic that this can be done, at least with the younger generation. All right, so with that, I think I will stop, and I will stop sharing so you can see my face and vice versa, but I'm happy to go back to slides as needed. So thank you.
So thank you, Thom, for a really thought-provoking talk. I'd like to open the floor for questions. Again, feel free to put them in the chat or unmute yourself and video yourself if you can to ask. And I'll ask one to kick things off while people are thinking, which is, I'd love to hear more about the device that you mentioned developing sort of pre-COVID in terms of field-based methods to get at inflammatory activation, because, as you've well shown and others as well, that, this one measure of CRP has, such limitations, and yet that's what we tend to be able to do in population studies. So how do you see that technology possibly driving the field forward?
Yeah, thank you so much for asking. And I swear I didn't plant that with Amanda, sorry. So I'm sharing my screen again. Can you see it? Do you see my screen?
Yep, we do.
Yeah, okay. So these slides I pulled out because I was worried about the time. But this is how we tend to study chronic inflammation. These are 4 people who've had their blood drawn every 3 days over a period of 2 months. And what you can see is that overall, an individual has the same level of CRP over time, on average. But there are spikes. There are spikes. It does go up and down. And then actually, the inflammatory system is very dynamic. Like ideally, you want to have basically no inflammation on your body. But if you get exposed to a pathogen, a microbe, a bacterium that is threatening the integrity of your body, you want that inflammatory response to come online very quickly. And that's what happens typically. It's called the acute phase response. So C-reactive protein is is effectively 0 or 1 or 2 milligrams per liter, but it will increase by 3-4 magnitudes, orders of magnitude within hours after an exposure. And that helps control the spread of the pathogen. You really want that online. But then you also want to turn it down because it can do a lot of collateral damage. So getting insight into these dynamics is impossible with a single measure, a single blood measure. And so the psychoneuroimmunologists and my colleague like Greg Miller get around this by drawing blood, stimulating it with a ligand like LPS. This is a bacterial cell wall product. It activates monocytes and other inflammatory processes. And you can also incubate a blood sample with a downregulatory signal like cortisol, glucocorticoids, to see how sensitive the cells are to turning themselves off as well as turning themselves up. And then you can measure the cytokine response or patterns of gene expression. Greg and colleagues have done this, in a lot of contexts by bringing people into the lab, drawing their blood and showing how, for example, low early life socioeconomic status leads to higher levels of IL-6 production when you stimulate cells. Well, here's what happens in the lab. You have to make your culture tubes in advance. You got your venous blood. You got to transfer the blood to the culture tubes. You got to incubate in this big incubator, 37 degrees. Then you got a centrifuge to pull off the serum, and then you aliquot and freeze. We just did a miniature version of that. So we take these tiny little culture tubes and we preload them with LPS and glucocorticoids. We draw finger stick blood with a capillary tube that has a fixed volume of blood, so we know how much blood we're putting in the system. We transfer the blood over. We found this portable battery-powered incubator, which is used, I think, primarily for veterinary purposes, like bull semen and eggs for in vitro fertilization, that kind of thing, out of Wisconsin, won't surprise you. And then we let that go for 4 hours in the incubator, which is set at 37 degrees, and then we transfer the contents over to a filter paper as a blood spot, which dries and preserves it. So we can do this virtually anywhere. And we were ramping up to go to the Philippines the summer of 2020 to try this out. We couldn't do that, obviously. So we're looking forward to getting that back online.
Great, thank you so much for that. There is a question in the chat from Victor. I don't know, Victor, if you want to ask it or.
Yeah, that's a good question. So, Victor, I'm happy to have you unmute and we can talk about it if you want, but I'll just say my initial reaction is that I agree that these factors are important. There's outside of my expertise, and I don't engage a lot with those kinds of behaviors and aspects of personality, but I do. I mean, the goal of that model is to bring all levels of analysis together, at least within a framework. No study can measure everything at every level and analytically put it together, I don't think. So this is something that you or colleagues might want to feature, but that's not something that we've been featuring in our work.
Thank you. I thought that was a terrific talk, by the way. I'm retired now for a number of years, so I'm not likely to do anything like that. But I've been impressed at how much of public health is influenced by human behavior. We focused on human behavior like smoking and exercise and diet and all that, but human behavior towards others, like what government officials do, what corporate officials do, or wealthy people in general, as well as racists and so on. So I think if we could find a way to understand that, it might open up some new pathways.
Yeah, I agree.
Thank you.
Bob, go ahead.
Hi, Tom. Thanks so much for just a great talk, as always. So I really appreciate it. So I really appreciate your kind of biosocial 2.0 challenging the biomedical model. And, I've thought about this a lot over the years and talked to others about this as well. And, part of the way you're doing this is through better collection of biological measures and figuring out the pathways through which these social exposures, end up inside the body and so on. But then I also wonder, in the end, okay, we collectively, turn out these findings, and, does it still have the potential or maybe even feed into the kind of still the same model that, we, in the end, we need to fix what's ever going on inside the body no matter what caused it at the individual level. So, without major changes in kind of what NIH does, that they just, took till 2021 to declare racism a threat to public health. I mean, is there real hope here, or are we in some ways just talking to ourselves, but at the same time, do you really see a chance for us to change this model that's so ingrained into our society?
Yeah. I'm less optimistic on the issue of genetic determinism and sort of a soft genetic determinism, which I think I would call the examples I was giving you today, where people just really prioritize the role of DNA as a causal force. And you can, I hear this in colleagues here in my department and other departments, like they'll refer to something as like it's in their DNA, and what they mean is that like you can't change that. And so I'm less optimistic on that front. I think there are a lot of cultural, historical reasons why we want to privilege genes and that sort of determinism. So I think it's harder to swim against that. It's probably not impossible, but that's why I was enthusiastic about epigenetics and sort of instead of changing necessarily people's desire to understand a causal force, but change their idea of what the genome is. It's not just something that's fixed and inherited at birth, but actually can change in response to experience. So the last example I gave you was humbling in that regard. I am encouraged, though, more generally that biological measures can provide good data that helps reframe some questions and debates. And the thing that gives me most optimism, or an example that gives me most optimism, is related to biology, but somewhat independent of that. So there's been a lot of interest, obviously, in race-based differences in health outcomes, a lot of interest in discrimination and health outcomes, and a lot of great work over the last 15 years on perceived discrimination, interpersonal discrimination, and how that relates to biology and health outcomes. But there's been this move over the past probably 5 years, but I've seen it most quickly taking off the last couple of years, to structural racism. And the most powering examples that I've seen of that with respect to making change in population health have linked health outcomes and biological measures to make the case that this thing way up here at the structural policy level impacts people inside their bodies. I don't think you can make that same case without the biological measures or vital records. So I find some optimism in that turn and moving away just from everything at the individual level, but linking the biology to the more social structural level. So we'll see.
Great. Thanks so much. Yeah.
Elizabeth, do you want to go ahead?
Sure. Thanks, Tom. Great talk. Thank you so much. Hey, so, I'm all for putting biomarkers into surveys as much as we can. Two things. I mean, one, we have to convince our respondents that they want to do it, and that, depending on the context, is not a small ask. And the other thing is we may not be able to afford to do it for everybody in this study, depending on the size. So, I guess I'm curious about your reactions as to what you found sort of the most effective in explaining somewhat complex tests in different contexts, and what, how you can help people understand why this is a good thing to do. And then the sampling's a different question, but
yeah, that's a great question. I mean, there are real costs, both in the cost to your budget, but also to time and training and outreach and logistics, and then potential impact on participants and their engagement. And I think that I just don't think there is one good answer to your question. I think it's going to be highly variable historically and socially and culturally. I mean, you and I have talked about our international work, and things are different in Cebu and Indonesia than they might be in the US. Ad Health, I think, has a really great track record of compliance with biological measures. Other studies have been less so. We just did a big study of seroprevalence study of COVID antibodies in Chicago. And we didn't do hardly any recruitment. The only incentive to participate was that we would tell you what your antibody result was. We had like tens of thousands of people wanting to participate in that study, and we limited it to what we could do with our budget. But that was a unique moment, right? So I don't think that's going to carry forward. So I don't think there's one good answer. I think having conversations with the people that you're going to do this research with about their perceptions and how they're going to interpret the findings is really important. So I would start there.
Linda, do you want to go ahead?
Tom, it's great to see you. Thanks
Hi,
for a
Linda.
wonderful talk.
Thanks.
My question is really about epigenetics and the timing of when the social factors and stressors in the environment are influencing the epigenome. I mean, the way that you presented the work makes it seem like those exposures throughout the life course can be changing gene expression. But the question is that the case, or is most of that epigenetic change happening early on in utero, and then it persists, and we're only measuring it later in life? So can you say more about the timing and the persistence of those epigenetic changes?
Yeah, that's a great question. And that study that we designed, which really was about 5 years ago when we started to put that in motion, is incredibly simplistic, right? We're looking at extremes of socioeconomic adversity and not a huge number of people. And we do find these big differences, but we don't know exactly what the critical or sensitive period of exposure was. We think it might have been earlier in life, but we don't know for sure. And a lot of that work at that time in human social epigenetics was really excited about methylation in particular, but other epigenetic mechanisms as the way that the early experiences were remembered and had lasting effect on health later in life, right? So we know there are lots of studies showing that early life adversity leads to shorter lives and less healthy lives. What are the intervening mechanisms in epigenetics? People were really excited about that. And there were a lot of animal model research and some human research to suggest that there were these sensitive periods early in development, and exactly when that period was probably dependent in part on which system and when it was developing at a certain point in time. But a lot of the research now has suggested it's not that simple, and it's not that methylation can change later in life in response to environments. And there's also a lot of methylation action in the body that has nothing to do with environment. It's just about differentiating tissues from one another and aging or having them not age. So the story is much more complicated than I actually presented here and how we initially thought this would roll out. So I don't have a good answer to your question other than to say that I do think there's good reason to think that early life exposures will matter disproportionately to some systems, and epigenetic mechanisms will be an important part of that process. But we shouldn't discount the fact that there could be changes later in life as well.
And they can be captured with repeated.
Right. So very few studies do repeat measures, in part because, going back to Elizabeth's question, they're expensive to do. So very few studies have repeat measures, but that's something that's starting to come online.
Thanks.
So Penny, I think you're our last question for the day.
All right. Hey, Tom, that was a brilliant talk. Great to see you. I just was wondering, if you have any words of wisdom to our trainees, because, the trainees, I think, in the different disciplines of their training don't always get to have that human biology training that, that you and Amanda and Linda and I have. And so what's your best recommendation for sort of the cross in both ways, the people who are more biologically focused to get the structural training and the people who are more in the social area to get the biological?
Yeah, well, that's a great question. And I'm going to actually spend an hour with the trainees right after this. So
awesome.
I can take it a bit. But I think, I think you have to be very clear about what your goals are and then pursue opportunities to attain those. It might be that you want to just learn how to manage and work with biological data, in which case that would send you down a certain path. Or it might be that you want to learn how to produce those data, and you could come to my lab and spend a year learning how we develop and validate and apply these methods. So I think we want to be clear about what your specific goals are, and then seek that out. I love working with postdocs, especially from UNC. So if anyone wants to come do stuff here, we don't have a great training program like you all have, but occasionally we have postdoc funding, or students can bring their own, and I'm always open to have that shared experience. And I think that's if you want to really dig into the biology, you need to do something where you have some more in-depth experience. And similarly for the biomedical or public health or biology types, they would want to immerse themselves in a more social science space. So I don't think there was one clear roadmap. It's going to be dependent on individuals' goals.
Thanks.
I think that was a perfect segue question. So thank you again for a fantastic talk. And I'm sure the trainees are very much looking forward to talking with you too. So thanks, Tom.
Great. Thanks. Great to see you all, and hopefully in person before too long.